All right, everybody, since that was set up and presented and I identified myself, once again, I am Chase Bernardi, and today I will be presenting my topic titled Determination of Gender, Genes versus Hormones. Human sexual development is an extraordinary example of how genes and hormones interact to shape biology. At conception, our chromosomes determine the genetic foundation of our sex, typically XX for female and XY for male. However, those chromosomes are only the beginning. It is the cascade of hormones that follow which sculpt our internal and external anatomy into distinct male or female forms. In this presentation, I'll walk you through how genes and hormones cooperate to determine sex, explore the key hormones and structures involved, and finally discuss what happens when this process goes off track. So to start off, we're going to talk about the genetic blueprint, the SRY gene. So let's begin at this chromosomal level. Every human cell carries 46 chromosomes, including one pair of sex chromosomes. Like I said earlier, XX for females, XY for males. The Y chromosome carries a crucial gene called SRY, short for sex-determining region of the Y chromosome. Around the 6th or 7th week of embryonic development, the SRY gene activates the production of a protein known as testis determining factor, or TDF. TDF triggers the undifferentiated gonads, which at this stage can either become the testes or ovaries to develop into testes. In embryos that lack the SRY gene, as in XX individuals, the gonads naturally develop into ovaries instead. In short, the SRY gene acts like a biological switch that sets the course of sexual differentiation. Without it, the body follows the default pathway, female development. So to continue with this, we're going to talk about hormonal differentiation on the male pathway. Once the testes form, hormones take over and direct male development. Inside the testes, Sertoli cells begins secreting a hormone called anti-Mullerian hormone, or AMH for short. This hormone causes the Mullerian ducts, which would otherwise become female structures, to regress. At the same time, Leydig cells in the testes produce testosterone, which promote the growth of Wolfonian ducts, the embryonic structures that form the epididymitis, vas deferens, and seminal vesicles. Some of the testosterone is converted by the enzyme 5-alpha reductase into dihydrotestosterone, DHT. DHT is a more potent androgen that drives the formation of the male external genitalia, the penis, the scrotum, and prostate. Therefore, male development depends on a precise balance of AMH, testosterone, and DHT. A defect in any of these signals can interrupt normal differentiation. So let's now talk about hormonal differentiation on the female pathway. In the absence of the SRY gene, the undifferentiated gonads become ovaries. Because there are no Sertoli cells, AMH is not produced, so the Mullerian ducts persist and develop into the uterus, fallopian tubes, and upper portion of the vagina. Likewise, because there are no Leydig cells to secrete testosterone, the Wolfonian ducts degenerate. Without DHT, the external genitalia develop into the clitoris, labia, and vaginal openings. It's fascinating that the female development occurs by default. It does not require active hormonal input in the early development. It's the absence of male hormones that allows the female pattern to unfold. So when genes and hormones conflict. So what happens when these genetic or hormonal pathways don't align? Conditions known as disorders of sexual developments, or DSDs, arise when there is a mismatch between chromosomal, gonadal, and phenotypic sex. For example, in androgen insensitivity syndrome, AIS, individuals have XY chromosomes and produce testosterone normally, but their cells lack functioning androgen receptors. As a result, their bodies can't respond to testosterone, or DHT, and they develop a female external appearance despite being genetically male. In congenital adrenal hyperplasia, CAH, or in XX individuals, adrenal glands produce excessive androgens, leading to masculinization of the external genitalia. Another example is 5-alpha reductase deficiency, where XY individuals can't convert testosterone to DHT. They're often born with ambiguous genitalia, but masculize at puberty when testosterone levels rise. These cases illustrate how precise coordination between genes and hormones is critical to normal development. So integration, genes decide, hormones control. So when we take a step back, we can summarize this relationship in a simple chain of cause and effect. Genes determine gonads, gonads produce hormones, and hormones shape anatomy. The SRY gene initiates testis formation. From there, serotoi and Leydig cells produce AMH, testosterone, and DHT, the key players in male differentiation. When SRY is absent, ovarian development proceeds naturally, leading to female phenotype. Therefore, we can think of this in a way: genes decide, hormones define. The genetic code sets the stage, but hormones sculpt the body's final form. So in conclusion, to conclude, the determination of gender is a multi-step biological process that beautifully demonstrates gene-hormone cooperation. Chromosomes establish the blueprint, XX or XY, but hormones execute the plan, creating the physical characteristics we associate with male or female anatomy. When either genetic signaling or hormonal activity is disrupted, the result can be a wide range of developmental outcomes, highlighting the delicate balance that underlines the human reproduction. Understanding this process isn't just anatomy, it gives us an insight into endocrinology, genetics, and human diversity. Thank you for your attention. If you have any problems, you can reach out to me and I will be glad and able to answer them.